Hello, everyone. Welcome. Thank you for joining us today. My name is Jesse Chen. I am a senior research scientist and application specialist at Acadium Life Sciences. And so I'll also be your host and moderator today for today's webinar. So today's presentation will really revolve around how you start your cell and gene therapy workflows and how that starting point can really affect everything that you do downstream, right? So we'll discuss today how a consistent and well-characterized starting material paired with a gentle and scalable cell isolation technology can efficiently move your programs from initial PD all the way to your commercial scale manufacturing. So now I'll introduce our two presenters today. I'll give a brief introduction, and then we'll have each of them give a bit more background on themselves, as well as our respective companies. And then after that, we'll hop into the rest of the presentation. For those joining us live, please don't forget to submit any and all of your questions that may pop up through the webinar. We should have some time at the end of the presentation for a Q&A session to answer all of those. So now, without further ado, I'll introduce our presenters today. So speaking on behalf of Acadium, we have Bill Lloyd, who is Acadium's Vice President of Research and Development. And we're also very excited today to be joined by Courtney Noah, who is the Vice President of Scientific Affairs at BioIVT. So Courtney, do you mind kicking us off here, maybe giving a little bit more color on yourself, on your role, and maybe a little bit more information on BioIVT?
Sure. Great. Thanks, Jesse. And I want to say thank you to the Acadium team for inviting me to join the webinar today. As you mentioned, I head up scientific affairs at BioIVT, and I lead a group of scientists that's really focused on understanding scientific challenges that are occurring within the industry, working with our clients across all of the different research spectrums and making sure we're really tailoring those BioIVT solutions to the workflows and applications that they're doing. So just to start, I wanted to give a brief overview of BioIVT. We've been around since 1981, and we've grown into a global organization over those years with over 600 employees. We have an extensive network, and I think what's really particularly relevant for this discussion is our global donor center network and just the scale of collections that we do through there. We have the ability to support both research and GMP workflows, and from a practical standpoint, what that translates into is more consistent access to leukopaks, those repeat donor collections, and even thinking about fresh versus cryopreserved material to meet the various research needs of our clients. And so that infrastructure really becomes critical as we talk about controlling variability, which in some of the subsequent slides I'll cover. And then just about our portfolio itself, we support work from target discovery through clinical translation. That includes our immune cell products, but also our normal human and animal biological samples. We have disease state clinically collected specimens, and then also our ADME products and contract research services. But like I mentioned, we're going to focus more on our immune cell starting material, especially the leukopaks that we provide. Because regardless of whether you're doing cell therapy, assay development, process optimization, really understanding the quality and the composition of that starting leukopak sets the stage for everything you do downstream. And I'll hand it over to Bill, I think, to give an intro to Acadium.
Thanks so much, Courtney. And it's true from Acadium side. We appreciate you and our partners at BioIVT joining us today. Right, since Acadium has been established in 2014, we worked on developing our cell isolation kits. And a core fundamental idea was that we wanted to emphasize quality cells, which will only improve the downstream research. Right, emphasizing this idea that everything we do in the lab or to treat our patients is only as good as the starting cells that are collected and how they're treated during the isolation. And we're so honored to be joined by BioIVT, whose leukopaks and cell products we've used almost exclusively over the last 5 years, which have been fundamental to our research. At Acadium, we are based in Ann Arbor, Michigan, where we produce and manufacture GMP cell isolation products. Importantly, and I'll go into this later, but we emphasize negative cell separation products, right? Trying to take that quality cell and not touch it. Not only are we GMP, but we have also filed our drug master file in 2025. The future of cell separation really is microbubbles, and that is because we can emphasize that healthier cells will increase recovery, improve cell engineering, lead to greater potency and persistence of the cells, while also improving your workflows. We can integrate onto existing equipment or remove equipment from your workflow, can start from a range of materials, starting from tens of millions to 100 billion cells or more with the exact same process, which is completed in under an hour, removing some of the variability and constraints of several hour processes. And lastly, right, we have been developing microbubbles with the clinic in mind. You can see all of the standards that we are compliant with. We have completed a successful GMP audit against US and European standards. We can provide in-bag GMP reagents for clinical use. And while we are still currently in the process of amending INDs so that we can be a part of human clinical trials, it is important to note that we have done what is the second best, which has been part of a human T-cell allogeneic product for canines in partnership with Lea Labs and the University of Missouri, where several canines have been put into remission from cells isolated with Acadium products. And while I can't wait to tell you more about our cell isolation, the story of donor to dose really starts with the donor, where our partners BioIVT are experts. And so I'll turn it back over to Jesse to introduce Courtney's slides.
All right. Thank you, Bill and Courtney, for those introductions. And so, yeah, like Bill said, we'll kind of now transition into the bulk of today's presentation. Just a reminder for anybody joining us live, as we go through this presentation, as Courtney and Bill are presenting their slides, if any questions come up, please feel free to submit those. I believe the chat box should be on the right-hand side of your presentation window. So without further ado, I'll pass it back off to Courtney.
Great. Thanks, Jesse, and happy to get started. So traditionally, I wanted to focus on how donor selection has really been driven more so by availability and kind of looking at simple demographics such as age, gender, or BMI. But what we've done at BioIVT is kind of taking a step back and look, well, there's a lot of information that can be paired with those factors. Ultimately, they really tell us very little about how immune cells behave and what you can expect in terms of that variability, especially from a leukopak. So what I really want to focus on is performance is driven by biology, and specifically the importance of looking at biology related to immune phenotypes and immune genetics. So what I'll focus on is how characteristics like immune cell composition, HLA type, KIR genotype can all really directly influence that leukopak performance, and more importantly, how you can use that information to reduce the variability, improve your reproducibility, and ultimately to make better translational decisions in your research. So whether you're looking across cell therapy or immunology workflows, we often see three kind of core challenges that emerge. The first, just the access to donors. And so I really want to call out the need for a broad donor pool that allows you to select those right immune characteristics. If you don't have enough to start with, as you go down further and trying to do your selection, it really becomes a challenge. Like I said, then you become dependent just on availability. The second is, and I think this is really critical, is those data blind spots. Most donor materials can be under-characterized, and if you don't have that HLA typing, you can have minimal functional data. And ultimately what that leads to is difficulty in interpreting your results. And then finally is the quality and consistency. I think there's a big focus out there on trying to make sure that the yield is as high as possible, but really that isn't enough. And what matters more is looking at that viability, that purity, and that absence of unintended activation. If any of those vary, what happens is your downstream results become unpredictable. And that's really where the criticality of having those standardized leukopak collections, that controlled isolation workflow, really come into play in ensuring that you reduce your upstream variability before your experiments really even ever begin. So we think this slide does a really nice job of kind of tying together those important concepts and making sure we hone in on those donor to donor variability that can ultimately influence your outcomes. So even when the collection is standardized, and we'll talk about that, it's definitely certainly important. Donors are human, right? We all differ pretty significantly in a number of different ways. And so what that translates into is differences that we see in that immune composition, potentially expansion potential, functional activity. And so what I kind of want to point to is, rather than treating donors as just interchangeable, just minimal factors that you're looking at, you really need to hone in on that donor as your primary experimental variable. And so it becomes essential that you're controlling that variable as much as possible to improve your reproducibility. So for BioIBT, we kind of dug into that and wanted to see, well, what's the variability when we look across all of the donors that we collect from? And these are our recallable leukopak donors, and happy to kind of get into that more detail. Hopefully you'll get a sense of that as we go through the talk. But there's differences that you can see across donors, but also wanted to dig into what types of differences do we see within donors. And when we compare that, it's pretty striking to see that when we look across donors, we see very high variability, and actually kind of it is most prevalent when you look at NK cell levels, where we see the widest difference between donors. But within donors, and so that kind of speaks to that collection consistency and also just reproducibility within the donor themselves, we see much lower variability, and really the donors are able to stay pretty stable with their immunophenotypes. So the implication there is, if you switch donors, you start to introduce a lot of that biological noise. If you work with repeat donors, that's one way that you can reduce that variability. And really, again, just critical nature of those recallable donor programs, the consistent leukopak sourcing, and really allowing you to anchor your experiments in more stable biology. And as I alluded to at the beginning, I think historically or traditionally, there's been a big focus on demographics and what type of demographic creates that ideal donor. But what we see, at least from an immunophenotype or immune cell composition perspective, is demographics really aren't a great predictor. When we ran the analysis on our donors, we saw negligible impact related to gender or BMI, and really only a modest correlation even with age. And I would say that was predominantly where we saw maybe some improvements with the NK cell population, but again, very minimal, so I wouldn't really even call it statistically relevant. So what happens is, if you kind of put these criteria in place and don't really understand the biology, you're reducing your access to a potential larger donor pool that would be more ideal for your specific application. So it's really critical that you're focusing in more on that immune phenotype and genetic drivers. So that's where we'll transition a little bit to from thinking just about who is the donor to how their immune system functions. And this kind of transitions us into the conversation around HLA type, which is certainly a critical factor when you're talking about immunology. So HLA determines which peptides are presented on the cell surface, and this is just kind of a brief overview. I'm sure many are familiar with it, but wanted to give this background. It directly controls what the immune system can see. So when an antigen is presented by the HLA, it is recognized by the T cell receptors, and that interaction is what drives the downstream T cell activation and specificity. So the HLA type is what's defining antigen presentation, T cell recognition, and ultimately that downstream immune response. But it also goes beyond that as we think about what role it plays really in therapeutics or various treatments. So HLA plays definitely a critical role in allogeneic compatibility. Also, as you think about transplant rejection, this is where we want to make sure differences in HLA between individuals can really be a primary driver and what the outcomes are there. And certainly HLA can influence immunogenicity. So if a therapeutic target is presented in the context of a specific HLA and your donor doesn't express that allele, that's where you can have downstream assay failure, fail to detect that appropriate response. And this can have certainly major implications as we think about safety and then even clinical translation. So as we know, therapies are ultimately deployed in patient populations with very specific HLA distributions. And so if your preclinical models, the experiments that you're running don't properly reflect that, you can really create a disconnect between the experimental data and the clinical outcome. So HLA isn't just a data point, it's really defines what your immune system can see and how it's responding. So just kind of all doubling down on the importance and criticality of understanding HLA and the role it's playing. So how do we apply HLA data in practice? And there's a few key ways this can become actionable. First, you want to make sure that you can match donors to your target epitope. So if your therapy, like I was saying before, depends on a specific antigen presentation event, it's critical to make sure that you're just selecting those right donors who can actually present that epitope. Second, you can align your donor pool with your intended patient population. And so kind of want to direct your attention to the heat map here on the right, because it becomes a very powerful tool that we can use. It shows that distribution of HLA alleles across our bio-IVT donors that have also been immunophenotyped. And it helps you select the frequencies that best reflect your target population. And so at bio-IVT, we have over 900 recallable donors. All of those donors have HLA typing associated with them. So like I said, you can really get a good perspective of what that distribution looks like, kind of pair it up with the distribution of your target therapeutic population or disease patient population. And I think, like I said, that's really important information to have. And again, the broadness of that access is really, really important and valuable. And then third, just to kind of tie it together, you can reduce the variability in immune assays. And so I wanted to make sure that we were extending this a little bit beyond just cell therapy applications. And so with that, it's really important because, again, you can have control and you can also have diversity. So if you want to make sure there's consistency in one type that you're looking at and you understand the implications there, or if you wanted to run more of like a panel approach and see differences. So leveraging this data can really be extremely important in your upfront study design. So to drill down a bit further, as I mentioned, when you're selecting donors, it's important to also consider that allele frequency. As you can see here, some alleles are very, very common. They really provide that broad population coverage, while others we see are less frequent. But they can, even the rare ones are equally important, just in different ways. Sometimes they're more associated with specific disease, progression, or pathologies. just to call out one example, for instance, HLA-A0201, that's one of the most studied alleles. It's widely used in immunotherapy research. And like I mentioned, some of the rarer ones that we have are commonly associated maybe with different ethnic backgrounds or disease progression. So the goal is not just to select any HLA, it's really to make sure you're selecting that right HLA for your application, for your target population, and thinking about how that's translated across diverse groups. So next, beyond HLA and T cell biology, we want to also consider NK cell function. It's a critical focus within the cell therapy industry. And this is where KIR receptors come into play. So again, a little background on them. They recognize HLA class 1 ligands. And what's really critical here is the interaction between KIR and HLA. And that's what determines ultimately whether an NK cell is activated or inhibited. And that comes about through different combinations, which have downstream signaling outcomes. And those can lead to either strong inhibition, which would then be maintaining tolerance to self, or they could allow for activation and downstream cytotoxicity. So the functional state of an NK cell is not determined by the KIR alone or the HLA alone. It's really by that combination. And those combinations are what ultimately shape the NK cell education, their responsiveness, their cytotoxic potential. And again, these all tie into implications for certainly NK cell therapies, anyone working in the antibody-dependent cellular cytotoxicity space or therapies around that, and also transplant outcomes. So if we look at some examples of that, KIR HLA mismatches can enhance tumor killing, while others may promote tolerance. And just like what we see with HLA in T cell biology, that KIR HLA pairing is really a key determinant in the functional outcome of the NK biology. So you want to make sure that you're focusing in on that and hopefully explicitly profiling for that as it relates to your application area. And so to kind of tie it in specifically to those application areas, as you incorporate the KIR and HLA data, you can begin to stratify donors based on the application that you're working in. So some examples of that are CAR-NK therapies. You can prioritize activating KIR profiles that then ultimately enhance that cytotoxicity. In transplant settings, you may look for those mismatches that can help improve leukemia control. As I mentioned, we have ADCC therapies, in which case they may be looking for specific receptor profiles that improve that antibody-mediated killing. And then for discovery work, perhaps again, you want a more broad diversity that reflects or accurately captures the population variation. So instead of treating all donors as equal, you really want to start matching your donor genotype to the therapeutic mechanism and representing a more translatable intentional design. So this is a bit of a busy slide, but it's really just meant to be kind of an example. I wanted to propose in a way like a checklist. So as you think about different applications, hopefully you took away from this, there's really no one universal best donor. And so it's really dependent on the application. So you can start to kind of rank different criteria that's important to you depending on the space that you're working with. So for example, T cells, perhaps you're looking more at HLA and high composition of CD3 and CD4 versus if you're looking more in immunoassays, more of a balanced immune subset could be relevant to your application. And so there's just different selection criteria and ranking that you can put in place there. So again, it just means that selection becomes more application driven, and that all ties into the ultimate design. And so the leukopaks really become not just that starting material, but as you can see different factors in here, they're really engineered inputs that can be tailored to your experimental question. And I'm excited to present this slide because this kind of ties into the collaboration and partnership that we have with Acadium. And you'll see it really is a complete workflow, and you have to look at the variables at each point, because I started off talking about the donor and the variability that exists there. Then there's the collection process, right? And it's just as critical that you're looking at that collection process, understanding the quality that exists or doesn't exist there. And I think this example really nicely conveys that point. So this was data that was generated in collaboration with Acadium. They tested over 50 leukopaks from BioIVT as well as another vendor. And what we know from leukopaks and cell isolation processes is that if there's a high contamination of red blood cells, that can really interfere with all your downstream processes, whether it's the isolation process or even the ultimate activity of the cells and viability. So that can lead to a lot of problems, potentially even batch failures. So in this head-to-head comparison, what we see is the BioIVT leukopaks consistently contains less RBCs than the competitor. And so it's a really nice example of, and we have even more data to share after this of kind of what some of the downstream results are, but really kind of striking example of, I think, how important quality is for these applications and how seriously BioIVT takes it, and the work that we do to get these types of great purity. And so just to kind of tie into where that comes from, because it's not a simple thing to get that high purity. BioIVT, as I said, we've grown from 1981 when we were first established, and we have decades of blood banking industry knowledge. And so that's what goes into the collection process and all the the work that we do to optimize our apheresis collections. We have rigorous training. We have standardized collection protocols. We also partner very closely with the equipment providers. So, and just to call out, for example, we work closely with Terumo, who over the years has been helping us to analyze the data on our instrumentation and walk away with more optimized units. And so it's a continually evolving process. And I think that it certainly benefits BioIVT and what we see as the quality of our products, and also benefits our clients because of the information that we have and understanding how that works and how to optimize it. So with that, I'm going to close on my final slide, and hopefully just kind of you take away these key points here. As I was just speaking to, really making sure that you're demanding that collection quality, ensuring that you're starting with clean, highly viable leukopaks is critical. Also looking at from the donor side, profiling for that consistency. And so as you look at repeat well-characterized donors, that's where you're going to eliminate that as a variable. And then also incorporating, and I would suggest incorporating it early, is looking at that genotype for function, and just kind of tying it back to the broad donor base. The broader the donor base, the more opportunity and availability you will have to make those decisions and really pair that genotype with the correct function. And then, of course, align that to your application and make sure that that biology is paired with a therapeutic goal. And so I'll end it here. I think that kind of hopefully leads in nicely to the next part of our discussion, where Bill speaking about how some of that optimal isolation method can help preserve that biology and unlock the full potential of the starting material, not just from the beginning, but the downstream applications.
And thank you so much, Courtney, for emphasizing how important the sourcing and the profile is. As you mentioned, I will now talk about the isolation and the manufacturing. But I wanted to start with a couple thoughts that really underscore the bridge between the BioIVT and Acadium partnership. I think fundamentally we were connected on 2 points, right, which Courtney emphasized. One is that we need to reduce the variability in our collections, that we need to control the processes the best we can because biology itself has enough variation. And so we need to control that variability. And the second key idea is that we need to emphasize quality. When I joined Acadium, I started with this idea, right, that we know how happy cells are in a human body. And it should be the mission of all cell therapies to collect those cells from the human body with as little variability and as high of quality and impacting them as little as possible. We should go through our manufacturing process in the most gentle manner possible. And then we should get those cells back into the human body where they can do the cancer fighting or disease fighting that we've designed them to do. And so I really think it's that reducing variability and emphasizing quality that unites everything we're doing here. I want to talk a little bit about how Acadium isolations work, and then I'll show you some data that we have generated in our lab that really emphasize that quality. As we talk about reducing variability, we know that a standard in the field right now is positive selection. But there's several drawbacks to that, right? One is you have several forces like a magnetic or a shear force, which can alter the cells of interest. You also don't capture a full T cell population, right? If you are only capturing a CD4 and CD8 positive cells, you lose double negative cells, which might be something like a gamma delta T cell, which the field has shown could be very important to therapeutic efficacy. And ultimately, we also know that these can be longer processes that are touching the cells of interest. And so Acadium reversed that question and said, can we have a negative selection at scale where all the cells of interest, right, every T cell that our body believes possible, can we use those cells in our workflows in a truly untouched manner? And the answer is that we can. And so if we look at this as an instrument-free workflow, what I'm really emphasizing here is that no instrumentation is needed for the isolation. We know that every workflow is different, that there are many cell washing instruments and bioreactors which are critical to the performance of cell therapies in the field. But this figure emphasizes that no equipment is needed in cell isolation. Acadium started with a philosophy of could we actually do an isolation where the cells are provided, right? Can we do that isolation within a blood bag? And I'll show you how that's done in 2 slides. But before showing you, I want to emphasize how Acadium achieves the isolation. We start with a silica microsphere, right, or microbubble that has an air-filled core and a silica surface. We then functionalize that with a streptavidin layer. And through custom antibody cocktail development, we are able to target any and every cell impurity within your sample. Within this presentation, we will emphasize the work of T cell isolations. However, right, because we are based on a platform microbubble technology, we can develop and design an antibody cocktail for any other of your cell isolation needs. Now, in particular, when we're working with in-bag closed system isolations, right, we're able to perform these isolations in the bag the cells are collected. We do that by adding the antibodies directly to the leukopak bag. They will label our impurities. We then add in the microbubbles that will use the streptavidin-biotin interaction to grab onto the impurities. And then we do not need any instrumentation because just by gravity alone, we are able to isolate the microbubbles from our untouched cells in our subnatant. And you can see that on the right-hand side, where the light pink underneath is our subnatant. Our upper layer of white is our microbubble layer. And then we are able to precisely move the subnatant into any downstream workflow step. Last slide to emphasize why this is so important. The NIH wrote a publication where they compared positively selected products to those of negatively selected cellular products and noted that positively selected products actually had much higher rates of toxicity, which can modulate the potency and the toxicity of those CAR T cells. And so it has been our mission at Acadium to emphasize data sets in our lab as well as the labs of our partners to underscore why this quality, why the increased potency, how can we improve potency but also reducing batch risk? How can we improve the COGS of these workflows? So in collaboration with Dark Horse Consulting, did a workflow comparison where we demonstrated that an Acadium isolation can save over 40% overall costs on a workflow today and perhaps even more in the future. And how can we save time and complexity, which is not only important as we consider those technicians and teammates working in the lab, but also of critical importance for our cells, right? We know that if we can interact with cells in as short a manner as possible, that those cells will have higher viability and produce more potent and persistent cells inside of our therapeutics. And so in this particular webinar, I wanted to highlight a couple facets of data that underscore this quality point. The first thing that we did is we had noticed as we were looking at different leukopak vendors is that sometimes, right, cells work very well when they're received first overnight after a 24-hour from collection to in the lab use. But that doesn't always persist if there was a 48-hour wait or a 72-hour wait. And so it was really important to us, even though we typically are using cells within 24 hours of collection, really important that the cells were collected with the quality that they could be used after 48 or more hours. And so here we show a comparison of these isolations across two donors in this case, where you can see that 24, 48, and 72 hours, the purity, all very robust at about 95%. And at none of those time points, moving to the middle, was the yield altered, all above 80%. And importantly, as I only show one figure on the right-hand side, but important to underscore that the CD4 and CD8 ratios that were so important to us, that those did not change from input pre-isolation material to post-isolation material, right? Also important to note here that we call out CD4 and CD8s, but there is a double negative population that is preserved as well, which is really important as we consider the therapeutics. And so while we started with this question of what happens with aged apheresis samples, the second question that we could ask is, well, how does this isolation persist over all the recallable donors that BioIVT has, right? This was, as I said, several samples, but we were curious, how would that hold up over a much larger data set? And so here we looked at 55 different samples that were collected from BioIVT, look at the pre-isolation CD3 percentage all the way from about 10 to 50% in a very robust purity, on average above 90% over all 55 of those donors. And so now we wanted to get a little bit more specific. We know that purity and yield are important, but as Courtney emphasized, right now we need to look at things like our cells expanding, or even further, right? What is the memory phenotype, right? Are we preserving memory phenotype? And taking that one step further, are we increasing the potency or preserving the potency and the persistence of those cells? And so this was a second study that was designed where we looked at 3 different donors from BioIVT. First, looking at isolation purity on the left-hand side, seeing what we saw in the previous data sets, right? That gave us a lot of confidence. Seeing in the middle that our yield preserved, right? Getting above 60% in that 70% or more range, very on par with what we've seen in our partners' workflows, that is superior to the magnetic isolations. And then lastly, right at the simplest level, tracking just the cell expansion over a 9-day process in a G-Rex platform. And showing, right, essentially that we can saturate the cell capacity within those G-Rex within 9 days, right? All great signs that these cells are performing to the quality that we want. But that wasn't enough for us, right? We know, and the science is only building on this with publications even published last week that we're very excited about from some of the scientific leaders in the field, that emphasize that memory phenotype might be more important than any other parameter for CAR T effectiveness. And so as we look at this, this is one of those 3 representative donors, looking at the post-isolation phenotype and carrying that forward to day 3 and day 9, where we see an emphasis, right, an increased percentage of the stem cell-like memory phenotypes. And we believe that that is what will underpin the persistence and potency of the data sets that we have. The last bit of data that I want to show today, right, really will emphasize this persistence and potency effect, right? We know that one of the standards in the field is to do a simple potency assay, right, at the very end of your workflow, where you might do a co-challenge. Here at Acadium, we wanted to look at a repeat challenge with the IncuCyte live cell analysis platform. And so for this work in particular, we compared it to a small scale Miltenyi isolation with a CD4/8 positive selection, activated those cells with Transact, and then initiated this work 6 days after isolation. We co-cultured with Raji GFP Luke cells, and we repeat challenged them 4 times, with each round lasting 2 to 3 days. And if I emphasize one of the 2 donors that were analyzed, right, we show that over 4 challenges, our control wells grow very robustly, saying that our cancer line is present in each of the wells and growing as expected. Now, over the first 2 challenges, so this is about hours 0 to 72, and say 72 up to 150. Over those first 2 challenges, right, the first challenge, you can see that both the Acadium and the Miltenyi arm do a good job of suppressing the cancer growth as expected. Now, it's in the second challenge where the Miltenyi arm does not do as well suppressing, but the Acadium arm continues to kill cancer cells. And while the Miltenyi arm did not show any killing over the 3rd and 4th challenge, the Acadium cells were very robust in continuing that rapid killing that persisted during the full challenge. And it's this thought, right, that we have now replicated not only in our lab, but also with several of our partners, that have underscored that the persistence and potency of negatively selected cells, right, taking the gentleness and the quality all the way from the isolation, or I'm sorry, from the primary cell collection all the way through the isolation and ending with the manufacturing, that will allow us to reduce the variability in our cell therapy products while emphasizing the quality that we need. And so with that, we will end our presentation. And Jesse, I will turn it back over to you.
Awesome. Thank you so much, Bill and Courtney, for this wonderful presentation. Very interesting. And just again, reiterating much with like anything in life, but particularly obviously in our case for these cell and gene therapy workflows, how you start can really impact what the final product is, right? How you finish. And so again, thank you so much for taking the time and presenting all of this information and all of this data for the rest of us, right? And again, a big thank you to everyone who joined us live, who shared some of their time with us to go through this presentation. So again, appreciate everybody for taking the time out of your day to join us. So with that, we will jump over to a quick Q&A session. We have some time here. Apologies, let me get this all in order. And I guess maybe to start, maybe we'll start with Courtney. I think this is a question coming in for you. From a bio-IVT perspective, how do you support scaling from early discovery, early work into potential clinical manufacturing, specifically in terms of donor continuity?
Yeah. No, thank you for that question. So I guess our kind of story around that is we built out GMP capabilities. So we have our GMP compliant leukopaks, which are branded under the VivoStart brand. But I think kind of as you think about it from early discovery into the clinical phases, the ability to kind of do all of your early screening, identify that ideal donor or donors, like what the cohort may look like, and then kind of reduce that variability as you move to GMP, because you're not changing your vendor, you're not changing your workflows or your process. And essentially we're mirroring, even in the RUO phase, the same processes and optimization that we have in our collections with the GMP product. So I think that really helps set you up for success. And yeah, hopefully that addresses the question.
Great. Thank you, Courtney. So we'll bounce over to Bill, kind of in the same vein as in terms of GMP. Was mentioned briefly in the presentation, but the question is, have the Acadium microbubbles or have the Acadium kits been used in any clinical trials as of today?
It's a great question, right? Not yet, although we're working with partners to amend clinical trials for INDs right now, right? It's important to note that Acadium and our microbubbles were developed with the clinic in mind. And so we have preserved the idea that safety and regulatory compliance was a core part of our philosophy from the very beginning. We worked with industry-leading virologists and clinicians to make sure that all of the ex vivo safety testing was performed, right? We have done a variety of in vitro assays in small animal models. And then, as I mentioned in the presentation, right, that led us all the way to doing a large mammal study in partnership with Lea Labs and the University of Missouri, where T cells, right, human T cells isolated with Acadium isolation or microbubbles, were used to develop an allogeneic CAR T therapy that was successful in treating 2 canines.
Great. Thank you so much for that answer, Bill. Moving on. So I think this question is to Courtney, potentially a little bit broad, but I'll let you kind of take it whichever way you want. Can you explain the process involving the recallable donors?
Yeah, so I think I'll tackle that question just in terms of like what the process looks like, and I think some of the things that kind of maybe differentiated for bio-IVT. So as I mentioned, we have over 900 recallable donors, and what that means is these donors have consented to come back repeatedly for multiple collections, and what comes with those is I really like to look at those donors as our like very highly characterized donors. So all of that information that I was saying is so critical to understand in your donor selection process and how that pairs with your application. That's where you have the HLA typing, the cured genotype, all the viral status, CMV, EBV. We do all of the viral screening on there. And, I think the other thing to recognize is that while we do leukopak collections, our donor centers do a broader blood-based and other biological matrices collections. And so we can kind of tap into all of those other donors as needed to kind of pull them into this pool of recallable donors. And we have our own recruitment services that pair with that. So if there's a need to kind of find that more unique donor, well, they're already in our donor base, maybe not in our recallable group, or someone that we can reach out into the more general population and pull in, and even access through our clinical networks. So, kind of pretty flexible, I would say, pretty expansive, and really gives all of that necessary information to make sure that there are none of those data blind spots, like I was saying.
Awesome. No, that's very helpful. Thank you, Courtney. A question, well, I will ask this to Bill, but Courtney, feel free to chime in maybe with some of additional thoughts or from a bio-IVT perspective. But I guess I think the question itself mainly geared towards the Acadium side. From a Acadium perspective, what other kits are available, right? Obviously, we presented primarily T-cell isolation kits today. Could we maybe, Bill, if you don't mind, talking to more so the catalog that we currently offer, and just kind of go from there?
Absolutely. I appreciate this opportunity. So, right, as we all know, CAR T cells have formed the foundation of where the cell therapy field has been. However, that is now bridging beyond just CAR Ts and has for several years, right? Because we utilize a platform microbubble, right? We can design an antibody cocktail to target any cells that want to be removed, right? And we work with extremely strong antibody vendors who are able to provide us with very specific targets that we've demonstrated work very well, right? So we now have this wealth of experience on removing any cell type, which means we can leave any other cell type untouched for your negative selection. So we can do T cells, we can do negative monocyte selections, negative NK, we can do negative CD34 isolations, and are looking to expand that into T cell subsets like CD4 or CD8 or Treg, and any other workflow that you can imagine, right? We are a strong partnership-driven organization, like you can tell by us joining or being joined by bio-IVT today. And so not only do we like to partner with our upstream providers, but also with our downstream clinical partners. And so the best thing that you can do is reach out directly to tech support at acadium.com, and we would love to have that conversation with you.
Thank you, Bill. I don't know, Courtney, if you wanted to maybe give a potential bio-IVT perspective of if in terms of kind of what therapies that you're seeing, or maybe ones that are up and coming. I know you had in the presentation you had mentioned some NK therapeutics, right? So I think, you know, in that vein, if you don't mind giving a little color there.
Yeah, I would say maybe not so much. Certainly, we see different, you know, specific cell types. I think one of the things we're starting to observe is, you know, the shift from oncology, because we also play in the different disease state areas. Shift from, well, not from oncology, but expansion from oncology, I should say, into those autoimmune indications and possibly beyond that. But, you know, and that's where, again, thinking about HLA type and what allele frequencies are critical in those different populations is important. And I did just want to call out, I know, you know, in terms of partnership, it's not just on the Acadium side. I know that we have also utilized the Acadium kits in our lab. I used to be in the lab. I'm no longer. I wish I had the opportunity to experience it for myself, because I have done many cell isolations, and they're very tedious with the density gradient system. So, and I know our you know, lab managers have had tremendous success and found, you know, all the efficiencies that you're speaking to, Bill.
Awesome. Thank you, Courtney. I think so. I mean, just kind of coming up here on time, I think I will say, you know, any of the questions that are left outstanding, we will follow up, of course. So no worries there. I think, you know, any additional questions that you might have after the fact, please reach out to and obviously to Bill, to Courtney, to myself. I think probably the most efficient method, at least from the Acadium perspective, is tech support at acadium.com. And then I think on the BioIVT side, it would be customer service at bioivt.com. And I think, you know, Bill and Courtney's contact information is on the, should be available on the webinar kind of informational page too. So feel free to reach out to them with any questions. But maybe kind of to wrap things up here, it was just kind of I know we had just, you know, both of you, you know, nicely kind of described how, you know, our partnership together between Acadium and BioIVT has been so important, kind of to develop not only the work that we presented in this presentation, but just kind of our internal knowledge and really getting our technologies and moving the field forward, right? And so maybe as kind of, I'll kick it to each of you, maybe I'll start here with you, Bill, of just in general, like the idea of where the cell therapy field needs to go. Partnerships obviously very important, but maybe just to kind of talk a little bit to that about our perspective at Acadium, and then just kind of from there, like where we hope the field can go.
Yeah, I appreciate this. It's a great question, right? And I think one of the things, and rightfully so, that the field has emphasized is once a process is developed, how do we reduce COGS, reduce variability, reduce batch failures, right? And that is extremely important. But what that misses is how long some of these processes take to develop, right? How much money in R&D efforts have gone into just developing processes to file an IND. And so I think one of the things that our partnership with BioIVT has emphasized is, through working together, how are we able to take our common partners and move them into an IND submission or IND submission preparedness as fast as possible, right? Because fundamentally at Acadium, right, we believe that the more INDs that are enabled through partnerships like with what we have with BioIVT, that ultimately is what's going to move the field forward, but most importantly, lead to the best therapeutics for the patients that we are all trying to serve right now. Courtney, if you have anything to add, please.
Yeah, I mean, I think you touched on, I think, you know, you're touching a little bit before, but I think the thing that I often also hear is really about the challenges and scalability and like you mentioned, cost. And so I think making sure, if I think about how we tie into those, right? It is the efficiencies that you're gaining through these optimized cell isolation processes, and then really making sure early stage, and I think you do save a lot of money and create efficiencies early stage by making sure you're looking at the right donors. If you make those assumptions too soon, this doesn't matter. There's a lot of backtracking and maybe adverse events that come up down the line. So I think that's also a critical aspect to consider. And again, just tying back to the importance of that donor selection and availability.
Awesome. Thank you both for those responses, and thank you again for sharing your thoughts and answering all of these questions. I think we're going to wrap it up there, right? So again, Bill, Courtney, appreciate your time and taking on the presenter duties for both of our companies. But yeah, if in the audience, if there are any other questions, please feel free to reach out. We will follow up in a timely fashion. But thank you to everybody for attending today's webinar, and hope you have a good rest of your day. Bye-bye.
Thanks, Jesse. Thanks, Courtney.